Chapter 9, Muscle Contractility. Skeletal muscles derive their name from the fact that most of them are anchored to bones when they move. They're also under voluntary control and can be consciously commanded to contract. A skeletal muscle fiber is a multinucleated cell as a result of fusion from myoblasts in the embryo. A single muscle fiber is large and highly organized. Each muscle fiber contains hundreds of cylindrical strands called myofibrils, or fibrils. Each muscle fiber is a cable made up of hundreds of thinner cylindrical strands called myofibrils, seen in longitudinal section. Each myofibril is made up of a repeating linear array of contractile units called sarcomeres. Each sarcomere, in turn, has characteristic banding patterns that gives the muscle fiber a striated appearance. Myofibrils are by cytoplasm with They're separated by cytoplasm with intracellular membranes and mitochondria, lipid droplets, glycogen granules all inside. The banding pattern seen in the EM on this slide is the result of partial overlap between the two distinct thick and thin filaments. So each sarcomere extends from one line to a next called the Z line. From the Z line to this Z line, yep, and contains several dark bands and light zones. There's a pair of lightly staining I bands at the outer edges of the sarcomere. The more densely staining A band is between the outer I bands, and also the lightly staining H zone in the A band center. Densely staining M line lies in the center of the H zone. The I bands contain only thin filaments. H zone only thick filaments. And part of the A band on either side of the H zone represents a region of overlap and contains both types of filaments. So each myofibril consists of a repeating array of sarcomeres. Each sarcomere is a banding pattern that gives muscle fiber a striated appearance, which we already said. This diagram up top of a sarcomere shows the overlapping array of thin actin-containing or orange filaments and thick myosin-containing purple filaments. The small transverse projections on the myosin fiber represent the amyosin heads or cross bridges. Electron micrograph of cross sections through the insect flight muscle down here show the hexagonal arrangement of the thin filaments around each thick filament. So the thin are the I and the A, and the thick are the H and the A. In the sliding filament model of muscle contraction, the skeletal muscle works by shortening fibers or ratcheting them across the actin or thin filaments. The bands actually remain constant in length, but decrease in width as they cross each other. So the A band remains constant in length, the A and the I bands decrease in width, and the Z lines at both ends of the sarcomeres move toward each other. So all skeletal muscle operates by shortening. As the muscle fiber is shortened, and the A band in each sarcomere remains essentially the same length, and the H zone and the I bands decrease in width during contraction and eventually disappear. During contraction, Z lines in both ends of the sarcomere move inward until contacting the A band outer edges. Contraction is not the result of filament shortening, but rather the sliding over each other. Thin filaments slide toward the sarcomere center, results in the increase in overlap between the filaments. Thin filaments contain actin as well as two proteins, tropomyosin and troponin. The tropomyosin occupies the gap between the two actin molecules. The troponin molecules are in contact with both the actin and the tropomyosin. In addition to actin, the skeletal muscle thin filaments also contain these proteins. The troponin is a globular protein made up of 3 subunits. Each has a distinct important functional role. And they contact both, as I said before, the tropomyosin components of the thin filament. The other important protein here are the tintin molecules. They are thick filaments composed of several hundred myosins along with small amounts of other proteins. Oops, sorry. Got ahead of myself. The third most important one is the tintin molecule. It's the largest protein yet discovered in any organism. The tintin filaments are elastic and prevent the sarcomere from being pulled apart during muscle stretching. So this tintin molecule holds it together. They stretch from one end of the sarcomere at the Z line to the M band at the center on either side. Tintin molecules originate at the M line of each sarcomere and extend along the myosin filament, continuing past the A band and terminating at the Z line. It's highly elastic protein and stretches like a molecular spring. Certain regions within the molecule become uncoiled. Tintin is thought to prevent the sarcomere from being pulled apart during muscle stretching and also contains myosin filaments in their proper position within the sarcomere center during muscle contraction. Tintin gene mutations have recently been found to be responsible for a condition known as dilated cardiomyopathy, which results in death due to heart failure. And then there's one more molecule called nebulin that acts as a molecular ruler by regulating the number of active monomers that are allowed to assemble into a thin filament. So during contraction, myosin heads bend and sliding the thin filaments over the thick filament. Energy from ATP hydrolysis induces a conformational change within the head. And then the elongated myosin neck acts as a lever arm and sort of moves the actin filament over it. So the attached actin filament is going to slide along or be ratcheted along by the myosin head. And in fact, the distance it goes is farther than the length of the neck and the myosin, so it sort of appears to be throwing it further than it could, further than the myosin could stretch. So the neck causes the actin filament to slide much greater distance would otherwise be possible with the elongated myosin. Energetics of actin-myosin interactions work like this. First, the energy gets provided by ATPase activity in the myosin head. Activated myosin attaches to the actin, initiating the power stroke, and then the release of ADP is followed by binding of another ATP. Absence of ATP prevents dislocation of cross bridges, causing rigor mortis in dead bodies. So the interaction of myosin with actin and the chemical interaction of ATP go like this. First, binding of ATP to a cleft in the myosin head causes the detachment of the head from the actin filament. 2. Hydrolysis of ATP to a phosphate in ADP energizes the head and fosters the beginning in step 3. 3. Causes the head to bind weakly to the active filament. 4. The release of phosphate, which causes a tighter attachment to the myosin head to the thin filament, and the power stroke that moves the thin filament toward the center of the sarcomere. And 5. The release of ADP, which sets the stage for another cycle. All this is triggered at the neuromuscular junction with the release of calcium gradient, which moves the troponin out of the way so that the myosin head can bind to the actin filament. So contact between the nerve and muscle is called the neuromuscular junction, and the linking of the nerve impulse to the shortening of the sarcomere is referred to as excitation-contraction coupling. Action potential in muscles is propagated into the cell interior by specific apparatus called transverse or T-tubes to the endoplasmic reticulum, here called sarcoplasmic reticulum, which releases calcium into the muscle. Calcium is housed in an elaborate network of the internal membranes that make up the sarcoplasmic reticulum, or SR, which is smooth endoplasmic reticulum. When an impulse arrives by means of a motor neuron, it's carried into the interior of the fiber A along the membrane of the transverse tubule to the SR. When that happens, the gated calcium gates open, releasing calcium into the cytosol of the muscle cell. The binding of calcium ions to troponin molecules in the skin filaments leads to the events described in the following figure in the contraction of the fiber. These fibers contract simultaneously when stimulated by an impulse transmitted along the neuron. The point where the neuron axis terminus and muscle fiber make contact, as I said, is called the neuromuscular junction, and the junction is the site of nerve impulse transmission. Unlike within a neuron-to-neuron connection where the action potential stays at the cell surface, skeletal muscles impulse is propagated into the interior through these transverse tubules, and they terminate in close proximity to the sarcoplasmic reticulum, which is going to dump calcium into the muscle, moving the troponin out of the way so myosin heads can bind. So, in the relaxed state, calcium levels within the cytoplasm of muscle fiber are very low and below the threshold concentration required for contraction. With arrival of action potential at the SR via these T-tubules, calcium ion channels in the sarcoplasmic reticulum membrane are opened, and the calcium ions diffuse out of the SR compartment over the short distance in the myofibril. So in a relaxed sarcomere, the thin filament of tropomyosin molecules block myosin binding sites on actin molecules. The tropomyosin's position within the filament groove blocking sites is controlled by the protein troponin. When the calcium levels rise, the ions bind to troponin C subunit, causes another troponin subunit to alter its shape. So the protein undergoes a conformational change, which moves adjacent tropomyosin closer to the center of the filament's groove. This shift in tropomyosin position exposes myosin binding sites on adjacent actins, allowing the myosin heads to attach to thin filaments and cross bridges to form. At that point, contraction occurs, assuming ATP is available. Each troponin molecule controls the position of one tropomyosin molecule, which in turn controls the binding capacity of 7 adjacent actin monomers in a thin filament. When stimulation from internerving motor nerve fiber stops, internervating motor nerve fiber stops, the SR membrane calcium channels close, and excess calcium is then pumped back from the cytosol back into the SR by calcium ATPase. And that's how muscle triggers are controlled. If we look at non-muscle motility with the cell, we can talk about how all these fibers facilitate that kind of motility. Non-muscle motility is more varied, and the critical components tend to be present in a less ordered or more labial transient arrangements, and they assemble and disassemble, just like actin and microtubules can assemble and disassemble. Actin filaments, often working with myosin motors, are responsible for a diverse variety of dynamic non-muscle activities, including cytokinesis, phagocytosis, cytoplasmic streaming, vessel trafficking, blood platelet activation, lateral movements and integral proteins within membranes, cell substratum interactions and cell locomotion, even axonal growth, and changes in cell shape. They're typically restricted to a thin cortex, the actin microfibrils, a thin cortex just beneath the plasma membrane. So there's this cortical zone. There's an active cell region that's responsible for a number of these activities right below the cell membrane. That's where all these microfilaments hang out. So actin-binding proteins then affect the localized assembly or disassembly of the actin filaments. And this activity is responsible for phagocytosis, ingestion of extracellular materials, extensions of processes during cell movement, and constriction of single animal cell into 2 cells during cytokinesis. Actin-binding proteins have the following roles in cells. Number 1, nucleating proteins. They provide a template for adding actin monomers in the ARP2-3 complex and formin. Also control branch versus unbranched filaments. So the slowest step in actin filament formation is the first step, nucleation. Nucleation requires that at least 2 or 3 actin subunits come together in the proper orientation to begin formation of the polymer. As with microtubules, actin filament formation is accelerated by the presence of a pre-existing seed or nucleus to which subunits can be added. Several proteins have been identified that promote nucleation of actin filaments. The best studied such protein is the ARP2/3 complex, which contains 2 actin-related proteins. Once the complex is activated, the ARPs are thought to adopt conformation, providing a template to which actin monomers can add onto. Another nucleating protein, formin, generates unbranched filaments, like those found in focal adhesions and contractile rings of dividing cells. Number 2, monomer sequestering proteins that bind to actin ATP monomers and prevent them from polymerizing. These proteins, like thymosins, are proteins that bind to actin ATP monomers, often called G-actin, and prevent their polymerization. And they're called actin monomer sequestering proteins. Without them, nearly complete polymerization of soluble actin monomers into filaments is favored by conditions within the cytoplasm, so it's a negative control. Number 3, end-blocking or capping proteins that regulate the length of actin filaments, like CAPZ and tropomodulin. Tropomodulin. Proteins of this group regulate actin filament length by binding to one another end of the filaments, forming a cap that blocks both loss and gain of subunits. Number 4, monomer polymerizing proteins that promote the growth of actin filament, such as profilin. Probably promotes actin filament growth by attaching to an actin monomer and catalyzes the dissolution of its bound ADP, which is rapidly replaced with ATP. The profilin ATP actin monomer can then assemble into the barbed end of a growing actin filament, which leads to the release of profilin. Number 5, actin filament depolymerization proteins that bind actin ADP subunits for rapid turnover of filaments. An example is cofilin. Cofilin has 2 apparent activities. It can fragment actin filaments, and it can promote their depolymerization into monomers. They play a role in rapid turnover of active filaments at sites of dynamic changes in the cytoskeleton structure. They're essential for cell locomotion, phagocytosis, and cytokinesis. Number 6, there are cross-linking proteins that alter the 3-dimensional organization of actin filaments. Examples are filamin, vilin, fibrin. Filamin proteins are able to alter the 3D organization of an actin filament population. Each has 2 or more actin binding sites, so they can cross-link 2 or more separate actin filaments. Regions of cytoplasm containing such networks have properties of a 3D elastic gel that resists local mechanical pressures. Other cross-linking proteins, like vilin and fibrin, are globular in shape. They cause actin filament bundling into tightly knit parallel arrays in microvilli and in some epithelial and stereocilia projecting from the inner ear receptor cells. Bundling filaments together adds to their rigidity, allowing them to act as supportive internal skeleton for these cytoplasmic projections. Number 7, filament severing proteins. These proteins shorten filaments and decrease cytoplasmic viscosity. An example is gelosin, which binds to the site of existing filament and breaks it into 2. Gelosin was, it breaks it into 2. Number 8, membrane-binding proteins that link contractile proteins to plasma membranes. Examples are vinculin, spectrin, and dystrophin, or also called dystrophin. Much of the contractile machinery of non-muscle cells lies just beneath the plasma membrane. Usually attach microfibules to a membrane indirectly via peripheral proteins. The forces generated by contractile proteins act on the plasma membrane, causing it to protrude outward, like during cell locomotion, or invaginate inward, like during phagocytosis or cytokinesis. These activities are generally facilitated by linking actin filaments to the plasma membrane indirectly by attachment to peripheral membrane proteins, like vinculin and spectrin. So those are the different proteins that affect the way actin is polymerized, depolymerized, attached to membranes, and attached to itself. In the microvilli, specifically, these are structures that are typical on the apical surface of epithelia that function in absorption of solutes, like lining your gut and your mostly lining your gut and some of your other epithelial surfaces and mucosal surfaces, and kidneys as well. Each microvillus contains about 25 actin filaments that are maintained in a highly ordered arrangement by the binding proteins villin and fibrin. The role of myosin 1, which is present between the plasma membrane and microvillus and the peripheral actin filaments, remains unclear. Some types of cell motility occur solely as a result of actin polymerization and do not involve myosin activity at all. Some of these types of polymerization events occur in examples shown here. And they're responsible as force-generating mechanisms. And the one on the left shows cytoplasmic, and right, shows cytoplasmic streaming in Listeria. Myosin is not required. So in this case, an ACTA protein on one side of the bacterium recruits the ARP23 complex for actin polymerization. In a Listeria, shown here, it contains a surface protein, that same surface protein, ACTA, that's present only at one end of the bacterium. And when ACTA is exposed within the host cytoplasm, it recruits and activates a number of these host proteins. And then once the ARP23 complex is there, it starts polymerization that moves the cell forward without myosin. Recent studies suggest that the same events occurring during Listeria propulsion are used for normal cell activities, ranging from propulsion of cytoplasmic vesicles to movement of cells themselves. So cells lacking cilia or flagella move by crawling over a substrate, right? And this cell locomotion is required for many activities in higher vertebrates, including tissue and organ development, formation of blood vessels, development of axons, wound healing, and protection against infection. Cell locomotion also contributes to the spread of cancerous tumors. It's hard to see cell locomotion in bodies since migratory cells cannot usually be distinguished from opaque cells around them in vivo. So researchers usually study single cells in vitro as they move over the bottom of the culture dish, shown here. During this series of activation, the cell locomotion is sort of, it's described as like walking, but it's basically changing in shape based on this formation and depolymerization of actin within the cell. So you get a protrusion of a leading edge called the lamellipodium, and then adhesion of the lower surface of the lamellipodium to the substrate that they're attaching to. That gets mediated by integrins residing in the plasma membrane. And then movement of the bulk of the cell forward over the site of attachment, and that's accomplished by contractile force exerted against the substrate. And then finally, the cell, you see the cell is moved over and has reformed its shape, still attached to the substratum, but the old attachments have been severed and the rear of the cell has been pulled forward. Here's more cells that crawl over substratum, and more formations of lamellipodium. And the force generated in the lamellipodium occurs by adding actin polymers to the filaments and providing temporary anchorage or focal points for the cell. So stimulus of the plasma membrane triggers local polymerization of actin during these events and causes polarization of the cell and movement toward the stimulus. Major structural features of cell locomotion occur. Branch cross-linked nature of filamentous actin networks resides just beneath the plasma membrane of advancing lamellipodium. This is a succession of short actin filament branches with that ARP23 complexes attached to them. So that's what they need to promote polymerization. And you can see it in the EM by immunogold labeling. It's not that clear, but you can see it, sort of gold labeling on the edges. These P23 complexes are stimulated by WASP proteins that reside in the Y-shaped junctions where the newly polymerized filaments have branched off pre-existing filaments. As actin filament polymerization and branching continue at the very front edge of the lamellipodium, actin filaments are depolymerizing toward the rear of the lamellipodium. Thus, the entire actin filament array undergoes a type of treadmilling, in which actin monomers are added to barbed ends of the array at its front and lost from the pointed ends of the array toward the rear. The so-called Wiskott-Aldrich syndrome disorder with crippled immune systems because white blood cells lack functioning WASP proteins and fail to respond to chemotactic signals. So WASP stands for Wiskott-Aldrich syndrome proteins, right? And this family of proteins shares similar domain structure and are involved in the transduction of signals from receptors on the cell surface to the actin in the cytoskeleton. And if you fail to have these, then your white blood cells won't migrate to sites of infection in the body. Advancing lamellipodium and branch cross-linked filaments network that reside just beneath the plasma membrane, which we already said, and the traction of forces, and this is showing those branches, right? Traction forces occur at sites where the cell grips the substrate, and the magnitude can be calculated from the dynamic patterns of substrate deformation. The greatest traction forces are exerted just behind the cell's leading edge, where the cell adheres strongly to the underlying substratum. Focal complexes that form near the leading edge of the motile cell exert traction force and then typically disassemble as the cell moves forward. Protrusion of the leading edge is followed by the movement of the bulk of the cell. The major forces involved in cell locomotion are those generated at sites of adhesion that are required to pull or tow the main body of the cell forward. They're often called traction forces since they occur at sites where the cell grips the substrate. It's harder to see here, there's in red here, but you can see all these focal points in this cell. So this is the extended lamellipodium, all the focal sites, and then it's going to pull the rest of the cell forward as depolymerization occurs where the sites were before. So axons develop by a process of active outgrowth and elongation, which you can see here. The bulk of the axon shows little evidence of motile activity. It's just the tip of the axon, or the growth cone, which is highly motile and shows several types of locomotor protrusions. And you get these so-called microspikes pointing outward at the edge of the lamellipodium. See all the microspikes, right? And then you get filopoda elongations that extend and retract during motile activity. So the growth cone explores environment and elongates its axon, and the lamellipodium and filopodia of growth cone respond to the presence of physical and chemical stimuli. So basically, the function of highly motile growth is to explore environment and to elongate the axon along defined paths. In embryos, axons of developing neurons, for example, grow along defined paths following certain topographical features of substratum or responding to presence of certain chemicals that diffuse into their paths, like we talked about when we talked about EMC. The lamellipodium and the filopodia of growth cones respond to the presence of these physical and chemical stimuli, and causing pathfinding axons to turn toward attractive environment and away from repulsive ones. The turning behavior of the growth cone, in turn, is dependent on dynamic changes in the organization of the actin cytoskeleton, which we've just described. The advancing tip and culture neurons will make dramatic turns in response to a diffusible factor that causes the disruption of the growth cone actin network. Ultimately, the correct wiring of entire nervous system depends on uncanny ability of the embryonic growth cones to make the proper steering decisions that lead them to the target organ they must innervate. For example, neurons grow out of embryonic retina toward a specific region of the brain. They encounter semiphorins, a family of proteins that acts primarily to repel advancing growth cones. The axons may grow along a path that directs them away from this repulsive guidance molecule. Back to our embryonic model. So each part of the body has a characteristic shape and internal architecture that arises during embryonic development. In A, the spinal cord is basically a hollow tube. B, the kidney consists of microscopic tubules. And C, the lung, for example, is composed of microscopic air spaces. Many cell activities are needed for characteristic organ morphology development, including a programmed cell shape changes occurring largely by changes in cytoskeletal element orientation within the cells. Development of the nervous system is one of the best examples. Toward the end of the gastrulation in invertebrates, the outer or ectodermal cells, situated along the embryo's dorsal surface, elongate and form a tall epithelial layer or neural plate. The neural plate cells elongate as microtubules become oriented with their long axis parallel to that of the cell. After elongation, neural epithelium cells become constricted at one end and thus become wedge shaped so that the entire layer of cells curves inward. This curving of the cell layer occurs after a microfilament band assembles in the cell cortical region just beneath the apical cell membrane. The microfilament band then contracts, narrowing that end of the cell. Eventually, the neural tube curvature causes the outer edges to contact one another, forming a cylindrical hollow tube that will give rise to the animal's entire nervous system. Which is pretty cool in that nerdy way. So this is an electron micrograph of the dorsal surface of a chick embryo as the neural plate is being folded to form the tube. Those are all the epithelial cells, and the cytoskeletal is the actin shortening that we just described, turning them in toward each other. So, to recap, the cytoskeleton is composed of three distinct types of fibrous structures: microtubules, intermediate filaments, and microfilaments made out of actin, which participate in a number of cellular activities. The tubules are hollow tubular structures that are the largest, 25 nanometers in diameter. They're assembled from the protein tubulin. In addition to the cytoskeleton, form the part of the mitotic spindle, centrioles, and the core of the cilia and flagella. Three families of motor proteins have been identified and characterized: the kinesins, the dyneins, which move along microtubules, and the myosins, which move along the microfilaments. In most cases, kinesins and the cytoplasmic dynein move materials along microtubules in opposite directions. The nucleation of microtubules in vivo occurs in association with a variety of microtubule organizing centers. And in animals that are called centrioles, but they are different depending on the types of cells and located different places depending on the different types of cells. And not all microtubules stay associated with their MTOCs. The microtubules of the cytoskeleton are dynamic polymers that are subject to shortening, lengthening, disassembly, and reassembly. Cilia and flagella containing a core structure called the axoneme that is composed of an array of microtubules that support the organelle as it projects from the cell surface and provide the machinery for generating the forces required for locomotion. Intermediate filaments are rope-like cytoskeletal structures approximately 10 nanometers in diameter that depending on the cell type might be composed of a variety of different protein subunits capable of assembling into similar types of filaments. And remember, those are less prone to polymerization and depolymerization, but require phosphorylation for stabilization. Actin filaments, or microfilaments, are even smaller. They're 8 nanometers in diameter and composed of a double helical polymer of the protein actin, and play a key role in virtually all types of contractility and motility within the cells. The forces responsible for microfilament-dependent processes may be generated by the assembly of actin filament, or more often as a result of interaction with the motor protein myosin. The contraction of skeletal muscle fiber results from the sliding of actin-containing thin filaments toward the center of the individual sarcomeres of a microfibril, and is driven by forces generated in the myosin cross-bridges that extend outward from the thick filaments. Non-muscle motility and contractility depend on some of the same proteins found in muscle cells, but they're arranged in less ordered, more labile, and transient configurations. Examples of non-muscle motility and contractility include the crawling of cells over substratum and axonal growth.